Okay, so good morning, everyone, and good afternoon, and welcome to this first session of this year's Virtual Podium Asia Pacific. I'm Anne Ben from SLING, the Singapore Lipidomics Incubator, and together with Dr. Fana Pino, we'll be our host for this morning's session. Before we start the actual scientific session, I'd like to give a massive shout out to the organizing committee, especially the fabulous multitasking Tusi, who pulled this all together. Thank you very much, Tusi, for bringing us all together. Also this year, for the first time, we've got cash awards, and we have publishers and journals to provide awards, one award per session. And today's morning session is provided by the Journal of Mass Spectrometry and Advances in the Clinical Lab. So, Tusi, you want to progress to the next slide, please? The format of this virtual podium is that we start with a keynote presentation for 15 minutes, followed directly by a discussion. After that, we will have 5 lightning talks of 5 minutes each, followed by a joint discussion for all 5 lightning talks together. Please, everyone who listens, put your questions already in the chat, preferably addressing whose person this question is addressed to. And then we have the second keynote speaker, which today is David Wishart, followed by another Q&A and the award ceremony. So I think with that, we start our first keynote. And the first keynote is given by Professor Peter Michael from the Baker Heart and Diabetes Institute in Melbourne, Australia. Peter is primarily focused on understanding how the dysregulation of lipid metabolism is associated with those big killers, such as cardiovascular diseases, diabetes, and Alzheimer's disease. And he focuses not only on early diagnostics and risk assessment, but now also on the development of new modulating therapies for chronic diseases. And today he will talk about comprehensive genetic analysis of the human lipidome and an online VPAP resource. Peter, over to you.
Okay, thanks, Anna. Let me see if I can share. Does that come through okay?
Yes.
All right. Thanks, Anna. Thanks, Tusi, and the rest of the organising committee. It's great to be here today, even though here is actually in my house, but it's still nice to be talking to you all. Let me jump straight in. So as Anna said, we're interested in lipid metabolism, dysregulation of lipid metabolism and cardiometabolic disease. And I think most people would appreciate that there's a number of drivers of cardiometabolic risk, and that includes genetic drivers, but also environmental drivers such as our diet, our lifestyle, other environmental toxins or pollutants. And I think to a large extent, the focus has been on how these influence lipoprotein metabolism, particularly the clinical lipids, so total cholesterol, HDL and LDL cholesterol and so on, and how that then impacts on cardiometabolic risk. And so these have been very useful biomarkers for many years now. In fact, these are also good therapeutic targets, and particularly for the drugs such as statins, fibrates, and PCSK9 inhibitors more recently, the targets primarily being LDL cholesterol. And as I say, these have been very, very successful. But I think we're starting to appreciate now that it's not only lipoprotein metabolism that mediates the risk between these genetic and environmental factors and cardiovascular and cardiometabolic disease, but also the underlying lipid metabolism. And that's really the area that we're focused on. So to that end, we've been developing for a number of years now a plasma lipidomics platform at the Baker Institute to look at those individual lipid species. And we use LC coupled with triple quadrupole mass spectrometer in a targeted fashion. And without going into a lot of detail, the methodology as it stands at the moment can measure over 750 lipid species in around 13 minutes. And that short run time means we can profile anywhere from 100 to 10,000 samples in a single study. And so these large clinical and population cohorts have been very informative for our studies. The methodology, I just want to give an acknowledgement to Kevin Hun, who developed a lot of this methodology during his PhD. And Kevin spent a lot of time separating out the different isomeric and isobaric species of lipids using reverse phase chromatography and then characterizing those using a series of different experiments. So here, for example, on the left, you can see these two sphingomyelin isomers, so a D18-1 24-1 species or a D18-2 24-0 species that can be separated out and so quantified separately. And then he synthesized a number of lipid standards here showing some lysophosphatidylcholine species with odd or branch chain fatty acids that allows us to deconvolute some of these complex chromatograms that we get. So these studies that Kevin did really led to the formation of this methodology now, where we've got this 770 odd lipid species covering a range of different classes and subclasses. You know, some have only a handful of lipids, others have, 70 or 80 lipid species within a specific class or subclass. But it gives us a very broad coverage of the lipidome, at least as far as plasma lipidome goes. Now, so let me move on now to talk about our genome-wide association studies of the lipidome. And so this is, here we've used a prospective cohort from the Busselton Health Study, and this was done in collaboration with Eric Moses at the University of Tasmania. The Busselton Health Study was a longitudinal study. It actually started in the 1960s and been running consecutively ever since. We had samples from the 1994-95 survey of about from 4,600 adults. We had a lot of demographic and anthropometric measures, as well as a history of cardiovascular disease. Importantly, we had blood samples, and Eric had already measured clinical lipids and GWAS data from these blood samples. And we had linkage data to the Department of Health in Western Australia, which gave us follow-up data for about 15 years of heart disease mortality and morbidity. So we took those samples. There was about just under 4,500 where we had plasma samples available. We extracted lipids from 10 microliters of plasma and ran through our LCMS system. At that stage, we were measuring just under 600 lipid species in total. And so this generated a large data set, about 2.8 million lipid measurements in that particular cohort. And so with that combined lipidomic and genomic data, then we were able to do the GWAS analysis. And this was largely done by Corey Giles in my lab and Gemma Cadby in Eric's lab. And so in addition to the Busselton Health Study cohort shown here on the left, we had two smaller cohorts where we were able to use for validation of our GWAS analysis. So these were from another Australian cohort, the ABLE cohort, and a US-based cohort, the ADNI cohort. And we had smaller numbers in each of those. So with those three cohorts, then we could do GWAS analyses in each of the three cohorts independently, and so we performed that. We also performed a GWAS in the Busselton cohort where we adjusted for clinical lipids, either by standard adjustment or NT-CoJo. Sorry about that. And the reason we did this adjustment for clinical lipids is we wanted to look at and see whether we could identify SNP lipid associations that were independent of the clinical lipids, or the clinical lipoproteins, I should say. So with those analyses, then we performed a validation using a meta-analysis of the ABLE and ADNI cohorts to see whether we could validate what we see in the Busselton cohort. And then we performed a discovery meta-analysis where we combined all three cohorts together to give us a maximum power for new discovery of genetic loci linked with lipid species. And so all of these combine then to identify these new genetic loci and regions associated with circulating plasma lipids. Okay, and with those loci, then there was a series of downstream analyses that we were able to do, looking at overlap with previous studies, association with cardiovascular disease-related loci, the relationship between serum lipid species and cardiovascular disease using a number of different association methods, and finally, co-localization analyses between lipid species and coronary artery disease to try and get some causal inference between particular lipid species and disease outcomes. So I'll just take you through some of those data. We don't have a lot of time, so I'll go through fairly quickly. So the initial GWAS is summarized here, where we're looking at genome-wide significance by p-values less than 5 × 10⁻⁸. There are about 74,000 SNP lipid class and lipid species associations, and that was with all lipid classes, and most of the lipid species were significantly associated with a large number of SNPs here, nearly 9,000. Encouragingly, though, when we look at the strongest associations, we see that they're genes that we might expect to see. So SPTLC2 and DEGS1 are involved in sphingolipid metabolism. We've got ApoA1 and PCSK9 involved in lipoprotein metabolism. And then some of the FADs and elongases involved in fatty acid metabolism. So these are things that we would expect to see in these types of analyses. So that was quite encouraging. When we look to do validation of those initial associations using the ABLE and ADNI cohorts, you can see here when we look at the correlation of the T statistic from the Busselton health study with the T statistics from the validation cohort, we get very good agreement, an R-squared of 0.89 for this relationship. And so of the 74,000 SNP lipid associations, we could look at nearly 69,000 of those in the ABLE and ADNI, and about 98% of those were concordant, that is, showed the same direction of association. About 86% were nominally significant with a p-value of 0.05. And when we adjust that for multiple corrections, we're still at around 29% validated in that independent cohorts, which we think is probably about the level that you would expect given the smaller size of the ABLE and ADNI cohorts relative to the Busselton. And the discovery meta-analysis, where we had a more stringent threshold, 3 × 10⁻⁻10, to allow for the effective number of lipid species we were looking at, still gave a very large number of SNP lipid class and lipid species associations. Okay, so we went on then to define the loci and genes controlling lipid homeostasis using various clumping approaches and ultimately narrow this down to 737 independent genomic regions. And of these, 31% have been reported previously in 35 studies that we could identify, which left a large number of novel loci that hadn't been reported to be associated with any lipoprotein or lipid or metabolite measure. And then using this ProGen framework, looking at either a genetic prioritization or biological knowledge approach, we're able to annotate nearly 500 of those genomic regions with both the top-down and bottom-up approach, so identify candidate genes in those regions. Okay, so I think this is one of the most interesting results that we see here in a global sense. So we were able to look at phenotypic associations of lipid species with coronary artery disease outcomes in the Busselton health study. And you can see here the forest plot shows this complex association of both positive and negatively associated lipids with coronary artery disease outcomes. Then using linkage disequilibrium score regression, we could look at genetic correlations between the lipid species and the same coronary artery disease outcomes using the UK Biobank and cardiogram. So now we're using over 500,000 people to look at these associations. And we see a very similar profile. And finally, we could use a polygenic risk score for coronary artery disease and again see a similar profile between these two. But particularly the phenotypic and genetic correlations, when we look at the coefficients for those, we see again very strong relationship between the genetic correlation and the phenotypic correlation. And what this really is saying, I think, is that what we're seeing here in relation to the lipid pathways that are contributing towards cardiovascular risk that we identified with the genetic correlations, the phenotypic or environmental associations are really seem to be acting via the same pathways. And I think this is important when we consider modifiable risk factors for coronary artery disease. Okay, and so finally then we could do co-localization of lipid loci with coronary artery disease loci. And here's just a summary of that. There are about 43 loci where we could get co-localization of lipids, and some showed only localization with a single lipid and others with several or many lipids, particularly for the lipoprotein enzymes, regions here, ApoE and LDL receptor. But what we did see here was that hepatic lipase or LipC, there were 5 independent loci that associated with both coronary artery disease and lipid species. And so then when we looked at the genetic analysis of the LipC gene region, and we took the lead SNP there and looked at lipids associated with that lead SNP, we see this really strong signature for phosphatidylethanolamine and lysophosphatidylethanolamine species. When we look at the regional association plot then for, here it's come up, the regional association plot for total phosphatidylethanolamine and coronary artery disease, we see these very similar structures just around that LipC gene region. And then the linkage disequilibrium shows here that multiple independent SNPs in this region. And so this allowed us then to do this generalized summary-based Mendelian randomization, or GSMR. And so here we can see very good agreement or correlation between the effect size of the SNPs in this region against total phosphatidylethanolamine and with coronary artery disease. And so down the bottom here is the forest plot and the GSMR estimate from the meta-analysis, which shows a highly significant relationship between the effect on PE and coronary artery disease. So all of this data together really demonstrates a shared pleiotropy between the LipC gene, phosphatidylethanolamine species, and coronary artery disease, which suggests then that PE species or phosphatidylethanolamine metabolism may be involved in the causal pathway to coronary artery disease. So in summary then, we have the human lipidome is largely heritable. We identified a large number of independent loci, and this co-localization analysis demonstrates a shared causal effect between lipid metabolism and coronary artery disease. And of course, this is the tip of the iceberg really for these data. There's many opportunities to mine these data for new insights into lipid metabolism and disease. And so just in the last couple of minutes, I just want to highlight some of the data that we're now making accessible on our website at the Baker Institute, because we appreciate that. I think there's a lot of opportunities with these types of data and with the methodology. And so we've put our methodology up on the website so people can explore that, or the FeeWeb allows the people to access the genomic data analysis that we've done. And so just quickly to show you a couple of slides from the website, you can access our website and look at the instrumentation and chromatography and the integration, I think, importantly, of the individual lipid species. And so if you click on an individual lipid species from the MRM transitions, you get a chromatogram and an expanded scale chromatogram. And so here you can see for a ceramide species, the chromatograms are very clean, but in fact, for a lot of species, particularly the phosphatidyl cholines and other phospholipid species, the chromatograms become quite complex. And so having these chromatograms enables people to easily identify which species they're interested in. And here again, some more complex chromatograms. And so we list out each of these with the assignment of those individual peaks. And then for the meta-analysis of the genomic data, you can click onto these different sites. And again, this allows you to basically search for a lipid species and so generate the Manhattan plot of all the variants associated with a given lipid species. You can zoom into a regional association plot and see the genes in that particular region. And then you can click on particular variants and get the lipid association plot for those variants. And then there's links to other relevant databases. Okay, so I'll stop there. I just acknowledge people in the lab, particularly Corey, who did all our genetic analysis, and the collaborators that worked with us on these studies. So thanks very much.
Thank you very much, Peter. This was beautiful. You know, I think you're also one of the most generous scientists I know in sharing your methods and your tools. Thank you very much for that.
Our pleasure.
Why I don't get it. Why PE? And was it all species, or was it the class of PEs in general?
No, it's pretty much all species. So it's a lipid class effect, and we see it for PE and lyso-PE species. Why that might be, we still need to investigate this. And of course, these studies identify targets and identify associations and perhaps causal associations, but the biology behind that is not so clear cut. What we do know about hepatic lipase, though, is mostly it's been associated with triglyceride levels. And so that's been reported fairly widely, that hepatic lipase acts primarily on triglycerides. And so that's always been thought to be the main driver of the coronary artery disease risk. But in fact, we really don't see a genetic link to triglyceride metabolism at all with hepatic lipase, and it seems very strongly linked to the phosphatidylethanolamine metabolism.
So then would you think that the triglycerides then is lifestyle and the PEs is really genetics?
Well, no, but you can, the PEs also respond to lifestyle. And in fact, when we look at associations between triglycerides and different lifestyle factors or different disease outcomes, and in fact, PE, those two tend to go along the same. So for example, triglycerides are strongly associated with diabetes and insulin resistance, but so are PEs in the same direction. So they're both controlled genetically, but obviously the link with PE and hepatic lipase seems to be quite specific. But they're also both responsive to environmental and lifestyle changes, which is a good thing, I think, because it means that potentially you can intervene in that pathway fairly easily. Yeah.
Yeah, it's not a good public health message if we have to tell everyone, sorry, man, it's just all genetics. Whatever you do.
No, exactly. Exactly, yeah. Which is why I think that correlation that we see between, the genetic correlation with cardiovascular disease and the phenotypic correlation is so tightly associated that's really encouraging that all of these things that we see genetically, they all seem to be also associated with environmental factors. So again, encouraging that we can modulate those.
That's time, man.
Any questions? James?
No, that's time, Anne.
So you're all invited to either type your questions in the Q&A or raise your hand and unmute yourself, or type it and I read them out. It's all fine. Peter, is the waist to hip ratio better than BMI?
I think it does show stronger associations with cardiometabolic disease. So I would say yes. Okay. BMI, I think can be misleading in a number of settings, particularly, where you've got, now I think at a population level, there's a lot of young men that are bulking up with exercise. Yeah. So that boosts their BMI, but of course they're very lean with it. So that tends to skew things, but generally abdominal adiposity is probably more important in any event than just subcutaneous.
Yeah, cool.
Right, I think that's time then, Anna. I think we need to move on to our next speaker.
Oh, okay, sorry. Thank you again, Peter. And now we start our 5 lightning talks. And the first lightning talk is given by Jenkit Tan from the University of Malaysia. And he's gonna talk about profiling fecal lipidome and metabolome of the aging rat using LCMS, a preliminary analysis. Jenkit, over to you.
Thank you, Anne. Good morning from Malaysia. Allow me to share my screen. I hope you can see my slide. Yeah. Okay, that's good. So I'm presenting a preliminary analysis on the fecal lipidome and metabolome of the spread dolly rat of the male rat, okay, using LCMS at different ages. So the fecal samples were collected at different ages of 6, 12, and 18 and 24 months of age. And the polar lipids and the polar metabolite and lipid were extracted using the Forge method. And separately, lipids and polar metabolite, they are acquired by LCMS in an untargeted approach. And the data were processed using MS dial for lipidomic data and using a compound discover for metabolomic data. And we use a metabolic analysis for statistical and pathway analysis, as well as the enrichment analysis. So the data were acquired in a positive and negative mode for both the lipidomic and the metabolomic data. So we use the partially square discriminant analysis for to identify the metabolite or lipids that are changed with age. So so the score plot indicates that there is a clear separation for the lipid species and the metabolite according to age, especially for the positive ion mode. The red is for the 6 months and 12, 18, and 24 months, and so for the metabolomic data as well. So this is a summary for the lipidomic and metabolic study or data. So the mass spectra, as I mentioned, was identified by the MS dial and the compound discover for lipids and metabolite mass spectra. And we identified the compound based on the MS2 data matching to the database that are integrated in the MS dial and the compound discover. So more than 800 fecal lipid species has been identified to be significantly changed with age, and more than 100 metabolite has been in the fecal sample has been identified to be changed with the age. So based on the PLSDA data, the lipid species that are related to age include like sterol, like acetylglycerol, ceramide, and fatty acids. And based on the enrichment analysis, the enriched lipid species belongs to diacylglycerol, unsaturated fatty acids, LPG, branch fatty acids, and LPE. These are the top enriched lipid subclass. So based on the metabolomic data, we found that many of the pathway are downregulated with increasing of age. This including metabolism of amino acid, for example, biosynthesis of valine, leucine, isoleucine, biosynthesis of phenylalanine, thyroxine, and tryptophan, biosynthesis of arginine, and metabolisms of glutamine and glutamate. So this is a prelim analysis. It's more to be done. In summary, we have identified several fecal lipid species and metabolite that will differ by age. Further study, of course, are needed to validate whether they are suitable biomarkers for the aging, and we are also interested on the potential role of this lipid species and metabolite on the aging process. So yeah, that's all from me. So thank you for your attention.
Thank you, Jakin. We take all questions all together. So we right away move on to the second lightning talk, which is gonna be given by, do I pronounce your name correctly? Tarangi or Tarangi?
Yeah, Tarangani. Yes, thank
Tarangi.
you. Yeah.
And Tarangani is gonna talk about finding biomarkers for early pregnancies in horses. Over to you.
Yeah, thank you. And let me share my screen first. Yeah, hope you can see all well.
Yes.
Yeah. Thank you, VPAP Organizing Committee, for giving me this opportunity to present here today. And I'm Tarangani Rukmali Pereira, a PhD candidate from PRC Reproductive Science at University of Newcastle. So today my talk based on piece of my PhD work, where we are working towards to develop a blood test to diagnose pregnancy in the mare. Little bit of background to this topic. Getting the horses pregnant is the most important thing in this industry, so as the diagnosis of pregnancy. But there is still no such robust test to diagnose pregnancy using blood or urine in mare as such in humans. The earliest possible date of pregnancy diagnosis is around day 14, and it is still invasive. So through an earlier detection to that will help the industry to reduce the economic imperatives and to enhance the animal welfare. So we have aimed to differentiate pregnant and non-pregnant mare plasma as early as possible, particularly at day 7, non-invasively by comparing the lipid composition between these two groups. So as the methodology, the mass spectrometry-based non-targeted lipidomics started with collecting blood samples from mares at day 7 and day 14 and generating plasma. At day 14, we have confirmed the pregnancies and tracked to the term. This allowed us to confirm the pregnancies at day 7 for our study. Using a batch mode approach, we have sequenced 16 male plasma samples using QX-active mass spectrometer, and the generated data were analyzed using well-established bioinformatical pipelines. So the statistical analysis revealed 24 lipidians displaying significantly different abundances between these two groups. You can see from this heat map how remarkable these lipidians are upregulating and downregulating between pregnant and non-pregnant mare plasma. Moreover, looking towards the lipid ontology data, we have found this significant cluster of enrichment with metabolism-related functions, which can be related with equine early pregnancy. Specifically, there was a lipid-mediated signaling activity, which has enriched with group of ceramides, which also has significantly increased in this pregnant mare plasma. So further pathway analysis revealed the relationship of these ceramides with several reproductive related hormone signaling pathways, specifically in progesterone. So I should say why progesterone is so important in this situation, because it is the key hormone in supporting the pregnancies in the animals. So in conclusion, this pilot lipidomics study has revealed with several lipidians, which is supportive in the early pregnancy physiology, especially in progesterone synthesis. And most interestingly, we could validate these findings by having our major proteomic study. We have used 264 samples and could reveal several proteins, which is significantly different in pregnant and unpregnant groups. And there was most interestingly the serpinase-6, which has significantly increased in pregnant male plasma, supporting the role of progesterone because it is the major transport protein for progesterone. So with these findings, we are planning to extend this study coupled with metabolomics as well by increasing the sample size and towards a better biomarker study. So with these novel findings, support the utility of mass spectrometry-driven multiomics for pregnancy biomarker discovery and indicate that systemic physiological changes occur as early as day 7 following fertilization in the pregnant male. So overall, the study represents significant progress towards establishing a panel of biomarkers for accurate detection of early pregnancy in the male. Lastly, I would like to thank all my supervisors, collaborators, and the funding bodies for supporting this project. Thank you for listening.
Thank you. Yeah, we take your question, for you later, and move straight away to the third lightning talk, which will be given by, apologies, I probably pronounced it wrongly, Xingtang from UPUS. Yeah.
Yes, hello.
Okay. Talking about short-chain fatty acids by GC. Over to you.
Okay. Right. Hi, everybody's good with the screen and my voice?
It's not the presentation mode.
Okay.
It's not in.
Yeah, okay. Go over to that side.
I mean, we could see it. It's fine. You can run it like that, or you click on presentation mode.
This is good now?
Now we see nothing.
I'm so sorry about that.
No worries. Yeah. Yes.
Yeah, but I have to go back in to do. It's still not in full mode, right?
No, but you want to click the full mode button at the bottom.
At the bottom.
At the bottom. Where's your cursor?
It's the bottom right next to the
Right,
scaling.
Yep.
So right to the left of that, to the left, right there.
Yep.
Right there.
This one.
Yeah. Mhm.
There you go.
Perfect.
Very much, guys. Thank you. Sorry for the hiccups.
No worries. No worries.
Okay, hi. So I'm Zing from Duke NUS Medical School in Singapore. Nice to meet all of you. So my lab is a metabolomics analysis lab, and we support researchers across many disciplines in the country with our facility. So we kind of churn up many kind of analytical panels to serve the researchers in whatever they are interested in. So today I'll be sharing on one of the analytical panels, the short chain fatty acids, the SEFA. I will discuss the methodology as well extraction optimization and the stability of SEFA in fecal samples. Yeah, so let me do a zoom in because the slides can be a bit small. Yeah, so a quick intro on short chain fatty acids. They are just the final products of fermentation by the gut microbiota when breaking down the starch and the dietary fibers in the colon. And they have received much attention because they affect human metabolism in a variety of ways. The major interests of the short chain fatty acids are the acetate, propionic, butyric, valeric, and their importance prompted an increase of papers over the last 5 years, as we tabulated over here. So the role of fatty acids has been shown to act as potential regulators in metabolic diseases, as well as mediators molecules in immunity and neurological functions. As shown in the pie chart, altered profiles of SEFA can lead to a wide variety of disorders. So the aim of this work is to actually develop a reliable analytical method and to establish the stability of the volatile SCFA in fecal samples. Here's a chromatogram showing the SCFA separation on gas chromatography triple quadrupole mass spec. The panel has since been expanded to include the minor SCFA and medium chain fatty acids such as the hexanoate and haptanoate. The quantitative parameters such as the linearity, limits of detection, relative recoveries are also shown in the table. And we have achieved good linearity and recoveries for all the SCFA here. As part of the extraction optimization, we examine the extraction efficiency and the temperature stability of the fecal samples in various conditions. So we established the extraction efficiency by the recovery of the SCFA in fecal samples. We found that a mixture of water and solvent, that is methanol, resulted in the best efficiency. The blue bars represent the mixture, the orange bars, the methanol, and gray bars, the water. We also compared the concentrations of SCFA from in fecal samples that has been homogenized or vortex. So to determine the kind of sample prep we will need for fecal samples. The results are not shown here, but the concentrations of the SCFA before using homogenizing or vortexing, they are similar, suggesting that the SCFA are readily soluble in solvents. Okay, after which we studied the stability of the SCFA in various conditions. The first study shows the collection of mice fecal pellets within 3 to 4 hours after production and after an overnight period. So the mice pellets were just picked up and stored in -80 straight, depending on the condition that it's being picked up at. The concentration levels we analyzed, we found that the volatile SCFAs has greatly reduced with the overnight collection, suggesting that the mice fecal should be collected ASAP. So to note that the mice fecal were collected from the same cage of from the mice. Yeah. So it should be representative. Okay, so this is an interesting one. We actually try to establish the stability of the SCFA in fecal samples for human poo stored in -80 over a half-year period. So in the upper chart here, it shows that the stability it's actually good enough to keep the fecal samples under long-term storage. Yeah, and then the last chart here. Sorry?
30 seconds to go.
Okay, the last chart is for the interest of the clinical study involving volunteers where the logistic chain may be a concern. So they can take hours to days to actually submit the sample. So we wanted to investigate the stability in ethanol and room temperature for 12 days. And actually the results looked pretty favorable, allowing ample time for the samples to reach the laboratory. So now I'm almost ending. So in conclusion, I actually showed a reliable method for testing SCFA in fecal samples, and it can be expanded to plasma. And hopefully the stability data that I've shown can be important consideration when doing sample collection. Okay, I'll be happy to answer any questions from the ground. Thank you guys for your time.
Thank you. Farhana, over to you.
Thank you, Anne, and thank you also for our first keynote speaker and also lightning speakers. So our next lightning speaker is Jeannie Yan. She's from University of Sydney, and she'll be talking about integration of metabolomics research into translational clinical care. And the example is cerebrospinal fluid biomarkers for neuroinflammation. So Jeannie, over to you.
Can you see my screen? Or...
Yes. You need to put it presentation mode. Yes.
Sure.
There you go.
Thank you for the introduction and good morning, everyone. In this talk, I'd like to take you on our journey of using untargeted cerebrospinal fluid metabolomics, which led to the preliminary identification of a potential panel for inflammation and then bringing this panel into the clinical space. Inflammation of the brain is increasingly recognized as important in acute inflammatory conditions such as encephalitis, but also neurodevelopmental, neuropsychiatric, and neurodegenerative processes. The high morbidity and mortality rates drive significant interest in understanding the underlying mechanisms involved in neuroinflammatory diseases. Here, we use cerebrospinal fluid metabolomics to explore mechanisms and facilitate biomarker discovery with the aim to offer earlier detection and potentially improved outcomes. So for the untargeted metabolomics study, a blinded approach was undertaken on a cohort of 14 acute encephalitis patients and 14 age-matched non-inflammatory neurological disease controls using LC high-resolution mass spectrometry. So here in the orthogonal partial least squares discriminant analysis score plot, we can see a clear discrimination amongst the two groups. Further, ANOVA and Fisher's post hoc analysis showed 3 metabolic pathways involved in driving the discrimination between acute encephalitis and controls. The first pathway we have is the tryptophan kynurenine pathway. So here we saw 9 metabolites contribute. There was statistical increase in kynurenine, 3-hydroxykynurenine, xanthranilic acid, anthranilic acid, and quinolinic acid with encephalitis patients, whereas a statistical decrease was observed in tryptophan, kynurenic acid, 3-hydroxyanthranilic acid, and picolinic acid. Notably, a marked decrease in tryptophan and increase in kynurenine levels led to an elevated kynurenine to tryptophan ratio observed in the acute encephalitis group. This ratio is generally used to infer the enzyme activity of indolamine 2,3-dioxygenase during inflammation. The activation of this enzyme causes the depletion of tryptophan and imbalanced formation of kynurenine pathway metabolites. Next, we have neopterin. This is a well-established clinical inflammatory marker, and we saw a significant elevation of neopterin in the encephalitis cohort, and this has been strongly supported by our clinical routine findings. In the nitric oxide pathway, we saw elevation in asymmetric dimethylarginine and arginosuccinic acid, and also a decrease in arginine and citrulline with encephalitis. Arginine and citrulline play an important role in the immune responses, and their availability during inflammation may serve as potential neuroinflammatory biomarkers. So from here, we propose that an inflammation panel, including neopterin, tryptophan kynurenine pathway, and nitric oxide pathway metabolites, may provide strong discriminative power for neuroinflammatory conditions, particularly in patients with negative neuroimaging and routine findings. So to successfully translate the research data, we needed to have a quantitative method. So to do this, we developed a method measuring these metabolites in 12 minutes on the LC triple quadruple mass spectrometer, and clinically validated the method in accordance with the NADA accreditation and International Organization for Standardization guidelines. This 13-minute metabolite inflammation panel was then applied to a cohort of encephalitis patients, an epilepsy group, and also compared against non-inflammatory neurological disease controls. So in the first year, we can see a distinct metabolic difference between the encephalitis and controls. We see a decrease in kynurenic acid, tryptophan, and arginine, and an increase significantly in neopterin and also 4 other tryptophan kynurenine pathway metabolites, more significantly an increase in kynurenine and quinolinic acid. The heat map for the epilepsy group shows a distinct increase in citrulline and kynurenine and a decrease in kynurenic acid. And a student t-test further indicated these metabolites were statistically significant. So our patient samples were run over a few months to show validation on the robustness of the biomarkers and reproducibility. As part of future work, we have larger cohorts phenotyped to demonstrate the sensitivity, specificity, and reproducibility of the panel. And more importantly, for a clinical setting, we'd like to define normative ranges. So in conclusion, we intend to deliver a time and cost-effective method capable of processing samples individually with a rapid turnaround time for translational clinical care. Thank you.
Thank you, Jenny. So as you know, our question and answer session will be all together in 5 minutes after we hear from our next speaker, who is Yi Yang. Yang is from Fudan University, Shanghai, and he will be talking about glycoproteomics, with comprehensive statistical control. So Yang, over to you.
Can you see my slide?
Yes, we can.
Okay. I would like to acknowledge the organizing committee to give me the chance to share my recent work on data-independent acquisition glycoproteomics. Large-scale profiling of intact glycopeptides is critical but challenging in glycoproteomics. The most common strategy for glycopeptide profiling uses the data-dependent acquisition (DDA) approach, with a major bottleneck that the precursor selection constitutes a stochastic element, resulting in missing value problem. To overcome this limitation, data-independent acquisition (DIA) have been proposed, where the instrument acquires fragmentation information of all precursor ions within defined isolation windows in a systematic manner. However, due to the increased complexity of DIA MS-MS spectra originated from multiple co-eluted precursors, quality control for glycopeptide identification is essential, but particularly complicated in DIA analysis. Herein, we propose GPRO-DIA, a pipeline that applies the concept of peptide-centric DIA analysis to intact glycopeptide. First, a spectral library of glycopeptide was built by DDA containing both peptide fragment and glycan fragment. Next, 3 types of device are generated, including glycan device with random mass shift and peptide device with reversed sequence, and both devices by performing the 2 operations successfully. Then chromatogram features of the target glycopeptide and the device are extracted from the DIA data. The extracted features were scored for the peptide part and the glycan part. Error rate of identification was estimated using a 2-dimensional force discovery rate approach, where the distributions of peptide and glycan scores were fitted using a bivariate 4-group mixture model. We further developed the glycoform inference algorithm to resolve potential interference of co-fragmented glycoforms. First, theoretical glycan fragment lines called identification transitions were generated in silico for each candidate glycoform when building the spectral library. Next, signals of precursors and identification transitions were traced and scored. The error rates of MS2, peak group, precursors, and identification transitions were integrated to the glycoform level error rate by a Bayesian hierarchical model. The performance of the glycoform level error rate control was tested on a yeast dataset using the entrapment strategy by adding false glycopeptides to the yeast library. The results suggest the satisfactory performance of error rate control both in the peptide part and in the glycan part with glycoform inference. GPRO-DIA is open source and available at GitHub. We hope this talk can help researchers to analyze the DIA data for glycoproteomics. Thank you very much.
Thank you, Yi. Now we are going to straight go to that question and answer session. So if you have any question, either you can put that in the chat function or you can unmute yourself and ask it. Before I open it up, I just have one question because I have seen at least a couple of you showing really good separation of your metabolomics or lipidomics data using a supervised statistical method like PLSDA and OPLSDA, but you did not provide any information. How did your model look like? So this question was probably asking Ginny and Jen Kit. So how was the model performance?
Yeah, maybe I can go first. We monitor the model by permutation tests available in the metabolic analysis up to 2,000 permutation. And we take the model if they are significant difference, that means the model is stable and reliable. Yeah. So we are solely based on the permutation test for the PLSDA. Yeah, I hope that answers your question. Yeah.
Yeah, we don't have enough time to go in detail, but I'm just giving you a heads up that, when you want to publish, that you'll have these questions, that, the feature you're going to select using how your model, they're quite related. So it would be important for you. How about you, Jenny?
So first we use Compound Discoverer to do all of the initial stuff. And then the next thing we use metabolite analysis. On our metabolite analysis, we selected some p-value cutoffs, and then we went into the actual raw data and picked up the actual peak areas. And then we had a biostatistician in our group at Sydney Children's Research, and then they helped us identify and think which ones were, and we saw these. So out of the whole few hundred detected, we only had 35 metabolites that were statistically significant. And then we looked at the clinical history of the patients, and we saw that there was the blood-brain barrier also included. So we thought metabolites that had an up and down regulation would more be able to represent neuroinflammation of the 35. I hope that answers.
Okay. Yeah.
Do you have a publication as well?
Yeah, all good. Thank you, Jenny. So I'm actually first going to take a question from one of the audience asking Yi Yang. The peptide shown does not have a consensus sequence for N-linked glycosylation. Can you please elaborate?
In fact, in my presentation, the peptide has a consensus sequence, and the sequence NXSOT. In my presentation, I denote the glycosylation N as J, so it has a consensus sequence.
Thank you, Yi. There are a couple of questions. James, how are we going with time?
One more minute.
Okay, so yeah, there are a couple of questions. I don't think we can take all of them. To Zing, do you know how similar SCFA profiles in feces are to plasma?
Hello, I'll be happy to answer that. The SCFA profiles in plasma is quite different to that of in fecal. They are actually very much lowered. The major SCFA we can see in plasma are the acetate, propionic, and butyrate, with acetate being the highest. So using this example, let's say I can find 10 millimolar of acetate in fecal sample, it's only 1 millimolar in plasma. Yeah, so that is much less detectable. So right now, we're actually doing a stability test on plasma that has been kept in biological fridge for like 15 years, and we'll be so excited. I hope I can share the results next time.
Thank you very much. I think I'll just take one more question that is from James to Jenkid. Is there a relationship between age and microbiome that could affect your results?
Yeah, actually, so far based on the metabolic study, we found one actually quite interesting pathway related to microbiome, which is the bile acid synthesis. And it was the pattern was increased first until the 12-month-old and starting to decrease the level after 18 months. However, we haven't started the 16S sequencing yet. We are hoping to correlate how is the gut microbiome related metabolite and the microbiome species as well. Yeah.
Thank you, Tim.
Farhana and James, if you guys wanted to stretch the questions a little longer, I think it's important for the students to have that. I can shorten my talk easily if you'd like.
No problem. Let's go to far past.
We actually had most of the question actually answered. I will just check again if there is another one. it was to Jen Kate. Again, how difficult of identifying branched chain fatty acid using untargeted analysis?
Yeah, surprisingly, when we have identified the lipidomic data, surprisingly, we found quite a number of fatty acids because the LCMS, for fatty acid, usually we are using a GCMS. And having that said, because we are actually using the database Lipid Plus in the MS-DIAL, so it is actually a putative identification. We hope to pick some of the most significant lipid species and do a validation with the authentic standard to assign the SN1 and the SN position. Yeah.
Thank you. David, if you have any questions to the students, please do feel free to ask.
Let's see. I'm trying to see. I guess it was for Ginny, actually, just a question about your triple quad measurement that you were doing in the cerebral spinal fluid. Did you guys have isotopic standards to quantify those?
Yes, so we wrote internal standards for almost all of them. So we have 10 internal standards for the 13 metabolites. So a few of the acids that eluted similar times would have shared one internal standard.
Know what your CVs were for in terms of reproducibility for those measurements? Were you around 10 or 20% coefficients?
For the low ones, we were 15% max, and for the high, the medium and the highs within 10%.
Okay, that's very good. Thank you.
Thank you.
Peter, do you have any question?
Actually, I have a question. Sorry. I have a question for the horses. So do I understand you correctly? The aim is to shorten the time it takes until you can detect the pregnancy, because right now you can only detect it, I guess, by urinary dipstick or something after 2 weeks, right? And your idea is to do it after 1 week, but right? And you call it an on-farm method, but I suppose you won't put a QE on the farm.
Yes.
And then what really is the time to results? Because you say you measure something on the mass spec, and then with data analysis and all of that, you lose another 1 week, right? So in the end, I guess, you've lost your 1 week of an advantage. Or is the idea to sort of translate the mass spec finding into something that is more easily done, dipstick type?
Yes, that's our main target is to do a robust test using you can use in the on-farm. So we use this mass spec analysis and everything to find targets, and these targets will be translated into the clinical practice as such in robust tests using any spec the ELISA test that can be performed in few minutes and get the results by the farmers itself.
Okay. Thank you.
Thank you, everyone. I think it's time for our next keynote speaker, Professor David Wishart. I don't think I need to introduce David. We have both of our keynote speakers are really generous, and you know that all the tools and they have been developing and sharing with us. And in case of David, you all know about MetaboAnalyst, and you know all the databases that he has developed, especially human metabolome databases, food metabolome databases. So David is talking about today in one of the topic, actually, I had a chance to collaborate with him 2 years ago to write a perspective article, multiomics integration. And so David, I'm looking forward to hear your talk, and I will just hand it over to you.
Thanks very much, Farhana. And thank you to Anne and Lucy and all the organizers for putting this together and for inviting me. I know it's morning there, it's evening here, so
evening here too.
To a close. Can you see the slides all right? So
Yes.
great. So as advertised, I'm going to be talking about some software tools and perhaps some databases to help with this concept of multi-omics integration. And I think something that was apparent through all of the presentations today is that we often have to use bioinformatics, cheminformatics, computational tools to try and bring these silos of genomics or proteomics or metabolomics or microbiomics or lipidomics together. And it's this smoothing out or roughing out the edges that allows us ultimately to describe the phenome, which is really what we're generally interested in biology. In all cases, whether it's metabolomics or proteomics or genomics, there's obviously lots of data that's generated. A lot of these are quite disconnected, and it's only in the last few years that people have really talked about how to integrate them. I think metabolomics is really well positioned among the omics disciplines because it's probably the most downstream of all of them. That is that genes code for proteins, which then manipulate metabolites, so that the changes at the genome scale profoundly affect the metabolome scale. And why, with data integration, we're generally able to consolidate and visualize that data. And I think that's a really important thing. And you've seen again with many of the presentations today how images, pictures, pathways are so important. It's also a way of converting a lot of raw data, basically things that we get in Excel files with numbers or digits, and converting that into sort of knowledge and giving some biological context. We often get lost in the data and forget that really what we're doing is to try and understand a biological system. We're often trying to get the macro scale interpretation, whether it's pregnancy for horses or cardiovascular disease and heart attacks with the molecular observables. And so I think this is why multi-omics is so important, because it links those silos together. It allows us to go from long lists to biology. And so this is often what we're confronted with on the left: lists of metabolites, lists of genes, lists of proteins. Sometimes we'll have quantitative values, sometimes we'll have qualitative estimates of them. But what we want are these pictures on the right. We want those pathways. We want those graphs. We want those charts. We want those biomarkers. So that, I think from my perspective, and Farhana mentioned sort of this review that we wrote a few years ago about the different ways of doing multi-omics integration. And you can parse it down into at least 6 or 7 different ways. There's probably more now. One is through pathway analysis. Pathways are a way of connecting, giving biological context to things. It allows us to link genes and proteins, SNPs and metabolites together in a map. Another approach, which isn't as organized, but is widely used now, is through network analysis. And we try and create either connectivity or semantic maps to connect concepts or objects together. Another approach, and this is sort of highlighted with Peter's work, and trying to look at multi-omic biomarker analysis. So sometimes we're going to combine SNPs with metabolites to come up with marker profiles, or genes with metabolites, or proteins with SNPs, and so on. That is an emerging trend for understanding or interpreting and getting biomedical knowledge. There's also an approach called ontology, ontological analysis. The gene ontology, or GO, is essential for a lot of protein and genomic analysis. We're working in an area called chemical ontology, or ChemFont, for functional ontology. There's also heritability, again, something that Peter presented in his talk about how we look at genomics and metabolomics and understand the genes and SNPs that contribute to variation in metabolite levels. And then another level is this genome scale modeling, where we use things like flux balance analysis and constraint-based modeling. So I'm going to talk about some tools that I've been involved with over the years, things that some of you are well aware of, others that you may not be, that help assist with this multi-omics integration. So one example is pathway analysis that you can do through MetaboAnalyst. Many of you have already started using version 5 of MetaboAnalyst. And so it's the most recent one, came out in April, May of this year. And of course, I think some of you already have shown some diagrams illustrating some of the pathway enrichment tools, pathway analysis, pathway topological, metabolite set enrichment tools. MetaboAnalyst 5 has been updated. It offers a lot of integrative tools, whether it's joint pathway analysis, biomarker analysis, functional analysis, network analysis. Some of the improvements for this year are better visualization, a more simplified interface. And as a rule, it's a really great resource for doing multi-omics integration. Most people don't realize that because everyone just uses it as a metabolomics tool, but it is designed and intended to help with that. And Jeff Shah, who many of you also know about, has worked very hard to extend MetaboAnalyst and has used many of the same tools originally in MetaboAnalyst to make other tools like Omics Analyst, which is a multi-omics tool, Microbiome Analyst, which also integrates microbial, genomic, metagenomic, and even metabolite data, and then Network Analyst, which was designed primarily for genomic and transcriptomic integration. Another tool that Jeff's group has developed and extended somewhat is something called OmicsNet. And this is, rather than using pathways, which you find in MetaboAnalyst and tools like KEGG or others, is to focus on network illustrations. So this one is also integrative. It combines gene expression, transcription factor, miRNA, and metabolite data, and allows you to create network models and images. Some of them are interactive, some of them are animated, and then connects that data that you provide with already previously known protein-protein interaction databases, metabolite protein data interaction databases, transcription factor gene databases to build out that network model. So this is a lesser known multi-omics tool, but one that I think offers really excellent visualization tools and something that I think more people need to be aware of. Something that I think not too many people are aware of is a project that we've been working on for almost 10 years. Originally it was called the Small Molecule Pathway Database, or SMPDB. It's been extended to something now called PathBank, which covers now almost 110, 111,000 pathways from 10 model organisms. This pathway database is intended really for the metabolomics community. It has a lot more metabolites than what you'll find in KEGG or other pathway databases. It has tens of thousands of lipids, thousands of other metabolites. It is supported in a variety of different machine readable formats from BioPACs, SBML, and others. The other part to this database is that the pathways are much more richly illustrated than what you might find in KEGG. They show organs and physiology, and they show tissues, subcellular compartments. They show the actual structures. They show the quaternary structure of the protein complexes. It's also able to capture a lot of information about metabolite signaling, inflammatory processes, immune responses that metabolites play a key role in. And this is something that we're trying to emphasize for the metabolomics community, that metabolites just simply aren't fodder for metabolism and catabolism. They are very important as key signaling molecules in the body, and often explain a lot of things that we tend not to understand. This is an example of how you can use MetaboAnalyst or rather PathBank to highlight and annotate increased or decreased levels of genes or protein levels, and to map those to pathways in a very detailed way. You can see some of the organs that are involved in this particular pathway, and you can tell by the color, red, orange, yellow, and green, the intensity and abundance of certain metabolites. Another area that we've been working on is in the area of biomarkers, and I think you saw at least 2 or 3 presentations today about biomarkers. There are protein biomarkers, there are genetic biomarkers, there are metabolite biomarkers, there are integrated biomarkers. And we've been trying to catalog a lot of that in a database called MarkerDB. This was released last year in a paper in Nucleic Acids Research. We covered a lot of validated biomarkers, and they include a lot of metabolite biomarkers, protein disease biomarkers, and genetic disease biomarkers. The bulk are genetic biomarkers, but in terms of the number of diseases that biomarkers covered, actually the metabolites win. There's hundreds often of mutations in a single gene, and those all count as biomarkers, whereas often it's just a couple of metabolites are associated with a specific disorder. So it's a rich database, has lots of information about receiver operator characteristic curves, sensitivity, specificity, biomarker panels, roles of all of these biomarkers in different conditions. And I think it's also a really useful tool for integration. Another area that we've been working on fairly actively is trying to develop an ontology for chemistry. Everyone, as I said, knows about the gene ontology. That's an ontology for genes, and it's been mapped to proteins for a long time. And that's how a lot of people do analysis of proteomics and genomics and transcriptomics. But in the world of chemistry and metabolomics, we don't have that kind of ontology. And so for the last number of years, we've been building out this ontology called ChemFont, or the Chemical Functional Ontology. It's trying to build out relationships so we can connect metabolites to biology, biochemistry, medicine, food science, and industry. We have information on the physiological effects, the disposition, roles, and processes of thousands of metabolites. And we've been working to make it quite compatible with the gene ontology and other well-known ontologies. So a draft version is now in the newest release of the Human Metabolome Database, which just came out last week. It's being upgraded continuously and is being expanded all the time. But what it does is it opens, I think, the door for people working in the field of metabolomics to start doing that data integration with terms, which is how a lot of multi-omic integration and analysis is done using things like ontologies from Go. An example of a resource that's still not quite ready, but is well on its way, is a database called the Microbial Metabolome Database, or MindDB. This is a multi-omics database that's trying to integrate metabolomic data, lipidomic data, proteomic data with microbiome and metagenomic data all in one resource. It captures information about the microbes, but also information about their effects on human physiology, health, the role that diet and bioactivity plays. We're using ChemFont to help integrate and visualize that data. I'll give you some examples, and we hope to have it released sometime in January or February. This is just an indication of the types of databases that we're integrating into MindDB. All of the small little boxes there, from BackMap to Knapsack to FoodDB to DrugBank to VMH. All of these resources are being integrated, compiled, and we're getting information, as I say, on metabolites, metabolism, bioactivity, health, microbes, medication, environment, diet. It's still under construction, and it's an enormous effort, but I think it'll be quite revealing and, as I said, quite integrative. There are different views to the database, where you can look at the host information of where you might find the particular metabolite, whether it's in the human urine, what levels it's found in, whether it indicates a disorder, the health effects of microbial metabolites, and their enzymes and their binding partners, and how some of these things were measured. There are the pathways, which are illustrated, and we use PathBank. There are also specific reactions for all of these metabolites, and both the microbial enzymes and the human enzymes that produce them and transform them. You can also look at the data through MindDB in a network view. So again, we're going from pathways to networks and back again. And this is a color-coded view that highlights how tryptophan will metabolize into a variety of compounds. And we learned a little bit about tryptophan metabolism earlier with Jenny's presentation, but also how it's metabolized in microbes to produce indole and indole acetic acid and indoxyl sulfate and indole pyruvate. We can also see what these different color schemes have. Some of these have sources. Some are found in seafood and beer. Some are found in cereals. Some are found in characteristic diets. Some have certain activities. Some will induce hypoglycemia, hypertensive conditions, pro-inflammatory. Others are associated very specifically with diseases. And so all of these connections can be drawn with this network, and all of them are obtained through this MindDB database. So it becomes a very rich resource for information, some of which you might have known, a lot of which you probably didn't know, all available through a network diagram. So I think we're seeing examples, and you'll see throughout the talks given the next few days with this conference, that there'll be lots of talks on metabolomics, proteomics, genomics, metagenomics, and that these need to be linked and integrated. Now, this can be done through a variety of programs and web servers, databases, and ontologies, and I've highlighted a few of them. A lot of them are freely available. We do our best to make them freely available, and a lot of them are very high quality tools that are cheap or essentially no cost. I think metabolomics is particularly good for doing this sort of integration, multi-omics integration, because it is the most downstream process. And people doing metabolomics have to basically understand a little bit about genomics, a little bit about proteomics, a little bit about metagenomics, and obviously a lot about metabolism. So with that, I just wanted to thank some of the people who've been working on these projects for a number of years. I mentioned Jeff Shaw, who's now at the University of McGill. Nama Karu has been working very hard on the MIME database. And some of our collaborators like Rima Kadir-Ook, Thomas Henkemeyer, Augustin Scalper, Vanessa Nico, and others, all who have helped build these resources and databases. So thank you very much.
Thank you, David. Before, I think I'm going to ask a question, because I think we have discussed this point before many times. This is a common topic of discussion in terms of, like, when you're talking about multi-omics integration in metabolomics, we still, in my experience, I'm doing plant metabolomics right now, and I cannot identify almost 80% or 70% of the metabolites. And when you're talking about pathway analysis or integrating with other omics, it becomes really difficult when we deal with so many unknowns. So do you think that we most probably need to go more in quantitative and also targeted metabolomics that could help with the integration better?
Yes, I think that's a really good point, that often to do integration from different types of omics fields, we do have to turn more to quantitative tools, quantitative methods. It allows us to relate values in one silo to values in another silo. So we've generally found that it's much easier to integrate quantitative proteomics with quantitative metabolomics. The numbers translate nicely, and we can see or map that are these real increases or real decreases, and are they significant? It's been a real challenge in fields, particularly in transcriptomics, where measures are more relative. And it's really difficult to try and integrate transcriptomic measurements into some of these other omics disciplines. Genomics is, sort of inherently quantitative. We can say there's a SNP, there isn't a SNP. We can talk about sequence. So that's more easily integrated, but it struggles with transcriptomics. We struggle with untargeted proteomics. We struggle even with untargeted metabolomics. To do integration. So, for now, most of the most successful examples of multi-omics integration have been with quantitative studies.
Thank you, David. I can see, Peter, your hand is raised, so please go on.
Thanks, David. That was a great overview. I wanted to draw you out a bit more on pathway analysis as well as Farhana. I think part of the issue with pathway analysis is often there's a lot of data that comes out. There's a lot of pathways that end up being identified. And I wanted to ask your opinion about assigning significance to different pathways and strategies to do that when you've often got multiple, associations coming into a pathway. Because ultimately you can either try and rank them in some way, or you can simply say, well, this is the one that we're interested in, or this is what, this was our starting hypothesis. And so we've validated that in a sense. But I think there's a danger that people see what they want to see in pathway analysis. And so if we can work more on developing, a bit more statistical rigor and getting significant values for different pathways, that might be quite useful.
Yeah, I think you bring up a really good point. And yes, there is that bias, particularly when people interpret pathways to sort of understand their process and that selection or self-selection. I think this is partly the reason why we've moved and been working really hard on this ontology idea. So, through gene set enrichment analysis, which sort of gave statistical rigor to gene set, it uses gene ontology data. It uses term frequencies, and it uses other statistical measures. and I think this way we might be able to help reduce that bias. And it may not be purely a pathway approach, but it still gives us that way of maybe rigorously identifying the pathway or rigorously identifying the processes, at least, that are important. So, I think that's where I can kind of see it going. Of course, there are methods in metaboloanalyst and pathway enrichment analysis that help sort of select the best pathways, ones that are most rigorously or topologically or statistically significant. But yeah, I think you're right. We still need to come up with more rigorous approaches overall.
Any other question for David? Please do unmute yourself and ask.
Hannah, I'm happy to ask one question, David. Great
Please
talk. Can I ask what are the availability of the isotopic labeling and flux analysis in cooperation with future studies in your? So is there any possibility with the carbon-13 labeling, or then we can easily guide the pathway?
So your question is about how we could integrate flux analysis into things like pathway analysis, or?
And is there any software development coming up in your group, David?
Yeah. In our group, not so much. Of course, we're part of a network in Canada. And so there is interest in both Jeff Shah's lab and also another lab in Victoria to do flux analysis. And they'll probably be developing some kind of software in the next year or so. So it's in the works, but I can't say that we're working on it. But hopefully, yes, it'll make more tools available for everyone.
Thank you.
Anyone else? Chris?
Yes, I have a quick question. First of all, David, great talk. Haven't seen you in a while. I think last time was in Salzburg, Austria. So great to hear what's been going on since then. I think this is really important stuff you're doing, and I didn't realize that these were extending into the multi-omic space. So it's great to hear that. What is the user base right now for this? First of all, I guess, how many users do you have on this right now? And I guess, what are you doing to try to extend that and communicate the value of this to the other communities?
Yeah, well, I think we've done some assessments. I think about something like 55% of all papers published in the field of metabolomics make use of metaboloanalyst or HMDB or one of these tools that we've talked about. So that's a fairly broad collection. We also know that some of our databases receive about between 10 and 20 million web hits a year.
Wow.
So they're used by a lot of different communities. I think metaboloanalyst has been used and is being used by people in the proteomics, transcriptomics, environmental communities. Other databases are being used by people in the world of pharmaceutical sciences. So they have a broad reach. And I think obviously there are ways of probably expanding the reach, trying to make them more user-friendly, more robust and rigorous. But yeah, we spend a lot of money trying to keep them up to date and to keep them running, and trying to improve them every year.
It's pretty impressive. And congratulations on that. Thank you.
Thanks a lot, Chris.
I think I'm going to take one more question before we go to the last phase of this webinar. I can see, Aaron, you have a question. Would you like to ask it by yourself? Okay, Aaron? All right, I will just read it out then. Okay, he's having a problem with microphone. That's okay. So Aaron's question was, running the MS is easier nowadays, but deciphering to get the meaning of it is challenging. Nice to see a comprehensive overview of the various informatic tools are being developed, making things easier. His question is, if we have an internal data, especially around compounds, is it possible to integrate this data into these pipelines?
Okay, I guess, I think the short answer is yes. If you have metabolite names that correspond to standard metabolite identifiers, those that are known, those can be generally handled through these software pipelines, whether it's MetaboAnalyst or OmicsNet or PathBank and others. If the compounds are completely novel and have never been identified before, it's still possible to use some of these things. And in fact, some of the software programs can be locally installed. So MetaboAnalyst can be locally installed. People have also been able to get versions of PathBank and other resources. So you can either use them through the web or you can have local installations as needed, especially if there's sort of proprietary data that you need to have. In the PathBank, we have tools like Pathways that allow you to draw your own pathways. And there is a community structure for people to deposit new pathways. And those are often now integrated into MetaboAnalyst. So they are connected. There are ways of doing this locally. There's ways of doing it through the web servers. But yeah, if you need help, just give us a call and we can try and help you.
Thank you, David. Jenkins, do you have a question? I can see your hand raised.
Yeah, thanks, David. I have a question. Thanks so much for developing the MetaboAnalyst. I have a question about the HMDB. Is that a possible, you are looking at the automation in the notation, compound identification, for example, we have hundreds of MS2 data, and we can, automatically detect the compound identification rather than we insert the MS2 data one by one?
So right now, I think with MetaboAnalyst or HMDB and also with CFMID, things are kind of structured to do one at a time. We are hoping to develop an API, so that's called an application programming interface for some of these tools, which will allow people to submit large numbers of queries. Those APIs will probably be developed over the next few months, but this would allow you to do sort of repeated requests and to try and do some more automatic identification. That said, a lot of commercial providers take the data from HMDB and CFMID and others, and so those are partly in those pipelines. So that's also there for you if you've got the commercial software.
Thank you.
Okay, good.
Thank you so much, David. We are almost at the end of this webinar, so I will be handing over to Anne, and thank you everyone. Thank you, Peter. I know you have to go. So I'm just handing over to Anne for the last couple of minutes and we are going to announce our winner of this session.
Yes.
Thank you, David.
Yes, exactly. Thank you, everyone. So now comes the award ceremony. And for this, I'm going to hand over to Chris Harold, who is in charge of the journal, JMSSEL, and he will quickly say a few words about the journal and then announce the winner.
Yes, I will. Thank you very much, Anna. Thank you for the opportunity to be part of this. It means a lot to me. Let's see if this is coming up. So first of all, to the organizing committee and to everyone who's been participating in this, it's really important. It's great to see that you're still staying connected and sharing science and doing science. If you don't know MSACL, MSACL is Mass Spectrometry and Advances in the Clinical Lab. We're a nonprofit association coming to you right now from Southern California in San Diego. We also have a journal of MSACL. So we've been offline like most people for the past 2 years, but we're back now coming this April, we hope and expect, in Monterey, April 5th through 8th. We have an abstract deadline coming up on December 15th for podium abstracts and February 10th for poster. So we're really looking forward to that. But what I'm really here for is JMSACL, also known as JIMSACL, the Journal of MSACL. We just recently were indexed on PubMed Central. It was a very exciting time. We're an open access journal. So if you submit to this journal, people can freely download and read your content. And right now we're investigating an innovative new way to submit and publish, which is pay what you can, where author fees are from $0 up to $900. So you choose the payment amount and get your paper published. So right now our site score is 3.2, and we have an impact factor that's pending. We expect when it gets awarded, it'll probably be around 2 to 3. So we're pretty excited for that as well. So I guess that's it for me on who I am and GMS-ACL. We'll get to the more important part of this point of why I'm here, which is the award. So congratulations to all of you Lightning presenters. It's really exciting to hear your work. And I've been told that the awardee for today is Jen Kitt. Congratulations, Jen Kitt. So as part of your award, I'll be sending you $250 in American dollars in cash. We prefer PayPal, but if you need some other method of payment, just let me know. But again, really glad to support this initiative. So thank you for taking the time to come and share your work, all of you. You know, it's always a hard decision when you're giving poster awards. You want to give everyone an award because you all had great content and great presentation presence. So thank you for doing that. But at the end of the day, you have to pick someone. And thank you again, Anna, for letting me come on and present, and for the relationship we've had since I met you 3 or 4 years ago at MS-ACL. It's been great to be on your team. So thank you very much.
Thank you. And thank you for generously sponsoring this award. And we follow up internally. And with this, I think we're coming to an end, and we can just again congratulate all of us for showing up despite a little bit of a Zoom fatigue. And thank you everyone for sharing your thoughts and the lively discussion. And thank you, Farhana, for co-sharing this. And see you in about 8 hours, 10 hours for the evening session. Whoever is available in the time zone.
Yeah. Thank you. Thank you, everyone. Thank you, David. Thank you, Chris. And
Thank you
everyone.
very much. Really enjoyed it. Well done, everyone.
Excellent talks, everyone. See you soon.
Bye for now.
Bye. Farhana, can you stay in the line?
Yes, Tusi, please stay.
Yeah.
So I'm going to take off, Anna, and we can talk later.
Yeah.
Okay.
Thanks, bye.
Well, sorry. Let me stop the recording.